So, thank you for the introduction. I'm Claudia Camarero. I'm a master's student at Universidad Autónoma de Madrid, and I'm going to talk about a research project I did under the supervision of Dr. Aida Verdes for my bachelor's thesis, which was called Differential gene expression analysis reveal potential non-predatory toxins in the heteronemertean Lineus longissimus. So, as many of you might already know, venoms are a rich and unexplored source of bioactive compound, which have shown a great potential in biomedicine and pharmacological research areas, as well as being very relevant in evolutionary studies, as the capacity of producing venoms is a key evolutionary innovation, which is a clear example of evolutionary. Even though the genomics field has been exponentially increasing these last years, there are still many taxa that are neglected, and one of these groups is the Nemertean worms, also called Venom worms. These organisms belong to the Nemertean phylum, which is comprised by approximately 1,300 species that are mostly marine endemic organisms. Their main characteristic is that they have an reversible organ called the proboscis, which is kept also characteristic of the organism's cavity called rhynchocele, and that they use it for predation as it is capable to secrete toxins. So they reverse the proboscis and they secrete the toxins. And in this video that I have over here, I hope it works, you can see how the ringworm, which is the long one, reverses its proboscis. There you can see the action it has on the prey, which is an annelid, as they feed on annelids and small mollusks, and it debilitates the prey, therefore facilitating its capture and then feeding on it. So apart from the secretions produced by the proboscis, they also secrete toxic mucus through their epidermis, which acts as a defense mechanism. But both of these secretions, the mucus and the one from the proboscis, are not produced by distinctive multicellular glands, but instead, stop the video 1 second, by secretory cells lying in the proboscis and voluble epithelia. So the study and characterization of these venoms becomes a task much more complicated because of that. So most of the studies until this day are focused on the mucus, and the majority of them are proteomic studies, while the proboscis remains poorly characterized. This is why in our research we have performed a differential gene expression analysis of the proboscis and the body wall transcriptome of the heteronemertean species Lineus longissimus, which you can see here, and specimen of this species. And the main objective was to identify and annotate upregulated genes in the proboscis, because due to the fact that it's the organ specialized in predation, they could represent putative novel predatory toxins. And how did we do this? Well, the first step was the sampling and RNA extraction. We studied 3 replicates of the body and 3 replicates of the proboscis. The next step was the generation of the cDNA libraries and Illumina sequencing. The thing with this species is that we didn't have a genome of reference to which we could map the reads by the Illumina sequence. So we first had to perform a de novo assembly of the transcriptome using all the reads, and that transcriptome is what we used as a reference. So the next steps were transcriptome assembly and mapping. We then estimated the abundance and we performed the actual differential gene expression analysis. And last but not least, we performed the annotation of the sequences based on the homology to already known genes using public databases. So our results showed that there were 516 genes that were differentially expressed. 141 of them were upregulated in the body, and 375 were upregulated in the proboscis. Out of the ones upregulated in the body, only 43% of them could be annotated thanks to the homology to what I just explained, to already known genes. And out of those, 4 corresponded to toxins, which mainly included the cytotoxin A3 and the alpha nematide, which are already known Nemertean mucus toxins, which had already been characterized and described in other studies, which so we were confirming what others had already studied and described. And because of them being upregulated in the body, they could be strictly related with the production of the defensive mucus, so they could have a possible defensive role. In the study, we focused more on the proboscis, as is the one specialized in venom production. So out of the 375 genes upregulated in the proboscis, 33% of them were annotated, and 24 corresponded to putative toxins. This 24 putative predatory toxins was classified in 6 toxin classes, which included mainly neurotoxins, cytotoxins, proteolytic lectins, as you can see in this alluvial diagram. And thanks to the homology to already known genes, we were able to infer the possible biological functions they could be playing in the Nemertean venom, which were mainly paralysis, cytotoxicity, and inflammation, which could help in the capture and digestion of the prey. So what I've just been explaining is the ones that were annotated, but we also looked at the sequences that were not annotated but are regulated in the proboscis, because as I've already mentioned, as it's the organ specialized for venom production, these non-annotated sequences would be of interest. And we identified 50 sequences that were upregulated in the proboscis. They had a signal peptide, so that meant that they were secreted, and they had similar testing patterns to conotoxins, which are potent neuropeptides from the common snail venom. And some of them also had interesting domains, such as the concavalin A-like domain, which has been seen to produce tissue, as an example. So these 50 sequences, we arrived to the conclusion that they could be possible novel toxins, but specific to heteronemertean organisms, because of them not being annotated yet, so they are not homologous to any other novel sequence yet. So yeah, they could be heteronemertean specific. So the main milestones and conclusions we reached was that we were able to identify defensive toxins, including previously characterized Nemertean mucus toxins, which validates our approach, as we are confirming the results of others have already obtained. And the main thing about the research is that we were able to study the diversity of putative non-predatory toxins with inferred paralytic, cytotoxic, and inflammatory functions, which will help in the capture and digestion. And even though our study expands our knowledge on ribosomal toxin diversity, we would still need to perform proteomic and functional studies in order to firmly confirm our results, which are studies that are currently underway. So this is the first part I wanted to talk about. And now I want to talk about a project that I've been part of after my bachelor's thesis, which is the microbiome. So what I've just been explaining are putative toxins that were identified, that were produced by the organism, as we have studied at an mRNA level. But there are some hypotheses and evidence that there are some toxins, such as the toxin and other Nemertean toxins, that could be produced, that their presence could be due to its production by commensal bacteria. So in this preliminary study, we wanted to assess the microbiome diversity in both the proboscis and the body to see if there were any differences. So the procedure was mainly, the concept behind the procedure is basically what I just explained about the differential analysis, but sequencing the 16S amplicon, which is specific to each bacteria species. So therefore, we were able to assess the microbiome diversity in the proboscis and the body. So you can see here in the right, these 3 first lines correspond to the body, and the 3 last ones correspond to the proboscis. And as you can see, the composition of the microbiome is really different. For example, in orange and pinkish tones, you can see gamma proteobacteria, and in the proboscis, the percentage is way higher than the body. And also, by studying the Shannon index, which is a measure of diversity, we saw that the proboscis had way higher diversity at the body. And the hypothesis could be that this could be due to the toxin production. And so then we studied that amongst the bacteria that were differentially present in the proboscis, which ones were related to toxin production, and from the body, well, same with the body, which ones were related to toxin production that were differentially present in the body. And the majority of them, for both of them, were gamma bacteria. So the difference that we found here, that is quite different, the diversity, does not really correlate with the toxin production. But this is a preliminary study, still ongoing. So I'll keep you updated with the conclusions we reach. Thank you for your attention. I'm happy to answer any questions.
Thank you very much, Claudia. So anybody has questions, so feel free to ask them directly or maybe just place it into the chat. And as long as we are waiting for any question, I might start with the first one. You talked about the cysteine pattern in the peptide you discovered.
Direction forward with the.
Can you repeat, please?
Okay. You talked about the cysteine bond pattern you discovered in the peptide sequence, and you said that they are equal or similar to that of the conotoxins. Did you also investigated the bond pattern of the cysteines, or just the sequence within the amino acids?
I don't really know what you're referring to. You mean like the not annotated ones that they had similar distinct patterns to toxins, right? Yeah, we classified, we just selected the ones that had the similar patterns, and then we wrote down which, because they are enumerated, so which one they represented, but we didn't get to investigate that. We just took note.